I want to welcome everybody and thank you for taking the time to be with us for this celebratory afternoon. We are absolutely hard pressed to believe that we have come to the end of the infamous summer of 2020 and are on the cusp of saying farewell, at least temporarily, to a wonderful group of students who joined us on June 1st for our virtual 2020 Gates Summer Internship Program, a program we all refer to as GSIP for short. We are delighted to be joined today not only by our interns, but also by our colleagues who served on the Gates Summer Internship Selection Committee, our Gates Center Director, Dennis Roop, and members of our staff, and very importantly, by some of our donors whose generous gifts toward GSIP have made the program possible. I want to introduce them to you in alphabetical order. I'm not entirely sure who might be on this call, and if I miss recognizing you, please forgive me, but Rhonda and Peter Grant, Mark Batrash and his wife Kelly, Missy Warner and the Glendorn Foundation, and Chris Wright. In any case, for those of you who may not be familiar with the genesis of GSIP, it actually began in the fall of 2014 when 3 Gates Center members, Tamara Terzian, Neil Box, and Enrique Torcia, approached our Gates Center Director, Dennis Roop, with the concept and a proposal for an internship program. At that time, there were only a few formal summer internship programs on the Anschutz Medical Campus, the most notable of which was at the Cancer Center. So these colleagues of ours identified a real need for undergraduates who were interested in cell biology, bioengineering, regenerative medicine, etc., in which they could find meaningful research experiences during the summer that were not solely focused on cancer. Dennis Roop was very receptive because the concept aligned perfectly with the Gates Center's priorities on education. With the help of Rhonda and Peter Grant and their amazing seed donation, the Gates Center launched the program in the summer of 2015 with 11 interns, a science lecture series, a keynote speaker, as we are doing today, and a poster session for the end of program activities. The subsequent growth of the program in breadth, scope, and impact is now history. Counting this class of 22 students that was chosen from 376 applications, we have accepted 119 students over the last 6 years. Key to our success in particular has been our wonderful GSIP team this year. Dr. Joe Brzezinski and I took over as directors of the program from our 3 founding colleagues, and we have had the great good fortune to rely upon the support of our program coordinator, Jessica Taylor Hurd, and our PhD volunteer, Jane Reck. Lucky us. And I would like to introduce my co-director, Joe Brzezinski. Thank you.
Thanks, Jill. Welcome, everyone. So as you can imagine, come earlier in the year when COVID-19 became so damaging to our ability to do research on this campus, we had to make some adjustments to our internship program. And of course, we wanted our interns to be on campus in our research labs doing full-time bench science, but that just wasn't an option due to safety concerns. And so we made some adjustments. And one of those adjustments was to host a webinar series throughout the summer for our interns to get to know the wonderful program mentors in the GSIP. And These webinars are actually recorded. I have a link down here at the bottom of the screen. It's a lot of text, but if you search for the GSIP program, you can find your way down to it. And so all the lectures that were done this summer have been recorded, and they're not all posted online, but they're mostly there. So if you're interested, you can watch presentations from any of the mentors who spoke this summer. Another thing we did was for some of our seniors who it's difficult for them to participate next year. We allowed them the option to do a remote internship and work with faculty, just not so much on campus doing wet lab research. And today we're going to hear from two of them, Ella Annest and Chiara Dart. And the other big adjustment we made is we allow for these interns to defer their enrollment to next year. And so really looking forward to having them on campus in 2021 to do their research in the lab instead of remotely. And so today our program is as follows. We're going to have two presentations from our interns, starting with Ella Annest in Dr. Fujita's lab, and then Chiara Dart, Dr. Shellman's lab. We're going to show you a short video that features our interns and their experience with COVID-19 and trying to do an internship. Then we're going to have a keynote presentation from Dr. Roop. Following that, we'll wrap up with some acknowledgements, and then after that, after our guests adjourn, we'll wrap up our summer program with our interns. Some business to deal with their last minute things. And so for our seminars today, I'd like you to hold your questions until the end, and then our speakers will field questions. And so I'd like to invite Dr. Fujita to the stage, so to speak, to share her slides and introduce our first speaker, Ella Annest from Berkeley.
Thank you. So I'm going to share slide. Yes. So thank you so much, and good afternoon, everybody. I am Mayumi Fujita. In my research lab, we study melanoma. So this cancer represents only a small portion of skin cancers.
Mayumi, I don't think your slides are showing.
Oh, it is not? Huh?
Yeah, we're not seeing them.
No? Ooh.
Try sharing again.
Okay, what happened? Oh, sorry. Okay, do you see it? Hi, do you see?
All good.
Okay, great, okay, thank you. Okay, so anyway, so I was talking about melanoma. So this cancer represents only a small portion of skin cancers, but it is responsible for the majority of death from skin cancers. So we are very interested in understanding why melanoma is so aggressive and how we can prevent metastasis, and also how we can treat melanoma patients better. We study tumor initiation, tumor heterogeneity, and also tumor inflammation and immune tolerance. Many projects to offer to students. But this year, because of the pandemic, we wondered how we can teach this talented student, Ella. Despite all our concerns, she took the most challenging project, a tumor inflammation project, and she made tremendous progress. We are so excited to hear her presentation today. Ella, take it over.
All right, can everyone see that okay? Great.
Yeah, looks good.
Thank you so much for the introduction, Dr. Fujita. So good afternoon, everyone. My name is Ella Annest. I'm a 2020 GSIP fellow, and I had the pleasure of working in the Fujita lab this summer. And so today I'll be sharing my work on the NOP1 inflammasome and its role in melanoma. So first, I'd like to take a step back and look at inflammation as a whole. As many of you know, inflammation is a really critical process that helps our bodies respond to infection, trauma, and other types of damage. And when properly regulated, it allows us to recover. But when it's dysregulated or occurs in the wrong time, place, or degree, it can actually augment that harm. And chronic inflammation is a known hallmark of cancer, and we're going to focus on inflammation in the context of metastatic melanoma, which is this horrible guy looking right here. So inflammasomes, and so this is an inflammasome complex right here made up of individual NOP proteins, which will be the majority of our focus today. So inflammasomes are really key regulatory proteins in immune response and help direct cytokine production. There's many different kinds of inflammasomes, and we're going to focus on NOP1, which isn't well characterized in the literature and does not have a solid crystal structure. And so previous work in the Fujita lab has shown that in metastatic melanoma samples, there are constitutively active inflammasomes that produce mass amounts of IL-1 beta, which is an inflammatory cytokine, which can then go on to induce autoinflammation that promotes tumor growth and will feed the melanoma. And so the lab's long-term goal is to elucidate the role of NOP inflammasomes in tumor progression. So this image here on the left is a single NOP1 sensor protein. And so when there's a proper immune trigger, kind of this diagram that I've made here, the NOP1 sensor will associate with the ASC adapter protein and the procaspase effector proteins. And so once these proteins associate, they can oligomerize and form an inflammasome complex, which we see right here. And so there's a bunch of different roles that the inflammasome itself will do, but it primarily will cleave that procaspase into caspase-1, which then has a key role in the maturation of IL-1 beta, which then has a really critical response in inflammation and other key cell processes. I also want to point out that NOP1 is special. So it has some extra domains that other NOP family proteins don't have, and that's the find domain here and the card domain. And so the find domain, we still don't know what the function of it is, but the card domain is pretty unique in the sensor protein because it allows the sensor to directly interact with the procaspase, and no other NOP protein is able to do that. So that's one area of research that we're excited to look into more. But mostly what I'm going to be focusing on is not the oligomerization of these NOP proteins that's essential for inflammasome assembly and further immune response. So the reason why we're focusing so much on NOP1 is because, as you can see here, melanoma has the highest mutation rates for NOP1 in patient data across all cancer types. So each of these little bars is a different type of cancer. And then if you look down here, you can see NOP1 expression in each of these red dots is a missense mutation. So compared to all these different cancers, melanoma is really interesting. And so additionally, we had a Cell 2016 paper that shows that NOP1 is the most prominent inflammasome sensor in human skin, and that all pathogenic NOP1 mutations are gain-of-function variants that actually promote inflammasome activation, which can lead to an increase, the mutations can lead to an increase in that self-oligomerization to form the complex when there's no trigger, and that will predispose individuals to skin cancer. So with that background and knowing that NOP1 mutations are an area of interest, we wanted to find out where these functionally harmful mutations were happening. And so to do this, we compared mutations from metastatic melanoma here to benign nevi, which are essentially moles. And so those served as our negative control. And so one of the first challenges in doing this was that while we know the different domains that exist in the NOP1 protein, we don't necessarily know where they are. And so using other well-characterized NOP proteins, we were able to do some alignments and some sequence homology and generate this protein map, which we were then able to input different mutational data to see if there were any hotspots or areas of interest. And so the melanoma data is from TCGA, and the nevus data is from one of our collaborators, Dr. Rick Sturm, who does work in Brisbane, Australia. And so the nevus samples seem to have more of a hotspot in here in the LLR region compared to the melanoma. And so that kind of suggested that the LRR isn't super critical or isn't implicated in that really critical oligomerization. However, we do see more of a concentration here in the HD1 region of that subdomain in melanoma, which is an area that we looked into further when we did more structural analysis. And so one thing is important to note is that not all of these mutations are equal. Just because you have a mutation doesn't mean there's necessarily a functional or biological change. And so to look more into that, we did some bioinformatic analysis and were able to score these mutations to get a better idea of what they were actually doing. And so I wanted to briefly go over how we sorted mutations to hypothesize which mutants might be more harmful. We used a variety of different scoring algorithms, which you can see right here, that measure damage based on sequence, structure, and overall homology, and then combine those individual scores to make a more weighted score that we were then able to use as a tool to see if there are any particular mutants that we could direct our focus on. And so here we have a graph of all of those mutations that we saw in the 2D maps with those bioinformatics scores. The yellow is the melanoma, the blue is the nevus, and it goes across the sequence overall. And these lower scores right here, so the more negative values, indicate that the mutation would be more damaging. And so you can see based off of the chaos of this graph that there really isn't a clear trend and that the scoring system didn't allow us to differentiate between the nevi and the melanoma. And you can see over here that the average score for each of them was nearly the same. And so it's important to note that one caveat of this scoring system is that it's dealing with individual proteins by themselves. We're more focused on the oligomerization and protein-protein interactions. So while we hope this would kind of be a preliminary tool to identify interesting regions, this wasn't super informative. So our next approach was to use survival data to see if we can analyze the effect of mutation on disease severity. So here we have a survival curve, and the red here is higher NOP1 expression, this blue is lower. And so we can see that there is a statistically significant difference between high and low expression of NOP1, and that lower expression of NOP1 is associated with poorer outcome. In these samples, we have both mutated and not mutated proteins, so the expression itself doesn't necessarily say if we have a functional protein. And when we compared the difference between mutants and non-mutants in terms of survival, it wasn't statistically significant. But knowing that low expression of NOP1 outcome, or that low expression of NOP1 results in a poorer outcome, we then looked to compare mutants and non-mutants in terms of their overall expression. And while we see a slight trend that mutants have lower expression, there's no statistically significant difference between the two. And so because of that, we wanted to explore further in the structural side of things, because simply because a protein is expressed doesn't necessarily mean that it's functional. And so because of this, we turned to more of a structural analysis to see if we could identify mutants on the protein surface that would likely interfere with proper oligomerization. So here was the part that I had a lot of fun with. So this is a model of the NOP1 protein. As I mentioned earlier, there isn't a crystal structure that has been published for NOP1, and so we were able to collaborate with Don Bacos here on the Anschutz campus. And so this is a really cool novel model of NOP1 that really doesn't exist anywhere else in the world, and here are just kind of various views of the same protein. And so once we got this model, I was able to do some electrostatic analysis of the protein, and so we can see that these red areas are areas of positive change, or positive charge, and the blue areas are areas of negative charge, and these charged areas are regions of the protein that we hypothesize are really critical in that protein-protein interaction. And the kind of white regions here are going to be hydrophobic surfaces. So those are really important for stabilization and kind of auto-inhibition of individual NOP proteins so that they don't necessarily oligomerize when they're not supposed to. And then the label domains that I have here are those subdomains that we saw in the protein maps before. And I also just want to point out quickly that the NBD region right here is also responsible for ATP binding. And so when this, when NOP1 is activated, this region will actually shift up. And so we think that even more of this charged area will be exposed, allowing for even more protein-protein interaction. So once we had that electrostatic model, we were then able to compare that to the mutational model. So all of this, each of these are exactly the same protein. This is the electrostatic model. And then we were able to map those mutations that we had seen earlier onto the protein surface. And so you saw a lot more mutations before, but just because there's a mutation doesn't necessarily mean it's on the cell surface or the protein surface. So many of the mutations are actually buried within the surface of the protein, and there's only a few that have the potential to be able to interact on the surface. And then the color coordination, or the colors of the different mutations correspond with the bioinformatics score. So red had the more damaging scores, yellow had the less. And so the Nevis model doesn't really show any mutants that we're interested in, because what we're looking at is seeing if there's any mutations that correspond with the hypothesized surfaces that we believe are interacting when these NOP proteins oligomerize. So primarily from this view, we're focused on this NBD region right here. And we can see in the metastatic melanoma area that this mutant right here is really interesting. And it's possible that it interferes with ATP binding, but also ATP binding isn't super well characterized within NOP1. So what we care a little bit more about is that protein binding. So here's another view of that same thing. You can kind of orient yourself by this image in the top right, but this is looking at that same mutant. So this is the NBD area right here. This is our area of positive charge. And we're looking particularly here, especially when this NOP protein is activated and this area shifts up and exposes this area more. Because this is a likely contact area, we think that this mutation is interesting. And then still in the Nevis model, we don't see any mutants that look particularly disruptive. And then this is just one final view. So again, orientation over here, this is looking at kind of the backbone of the protein. This kind of darker gray area is the HD2 region, and this other gray area is the LLR. And because we see such a negatively charged area, we really hypothesize that this is an important contact zone as we form that really beautiful complex that goes to be a functional inflammasome. And so looking at these two mutations right here, we can see that we're substituting a positively charged kind of smaller arginine residue with a nonpolar tryptophan. So we go from something that's charged to something that's uncharged. So we're having a chemical difference, but then we're also having a pretty significant difference in their overall residue size, which we think will just kind of have a more structural and geometric problem. And here again, we go from a charged residue to something that's not charged, so likely has some effect on chemical interaction. And then finally, we have one interesting mutant in our Nevis model, which is again, going from an arginine to tryptophan. So we have both that chemical and structural difference there. And so a lot of this work has been exploratory. So we have some things that work and other things that are like, okay, well, I guess we'll try something new. But overall, we have solidified that NOP1 inflammasomes are really important and highly mutated in melanoma. And so being able to better understand them has the potential to both improve drug development and patient treatment. We've seen that low expression of NOP1 is deleterious to survival, so functional NOP1 is hypothesized to have a protective role. And from those mutant models that I showed you earlier, we're continuing to explore that novel structure by performing mutagenesis experiments now to be able to show a clearer relationship between the structure and the biological function. And so with that, I'd really like to thank the Fujita lab, especially Dr. Jenny Sampson, who not only helped me through all of this, but finished her PhD in the process. And so thank you to the various funding sources that made this work possible, to all the incredible donors, and of course to the GSIP program for the opportunity to have this really wonderful experience. And so that completes my talk, and I'm now happy to answer any questions that you guys may have.
Thank you, Ella. Great job. We only have time for maybe 1 or 2 questions, especially from interns. If you have questions, please unmute and let them flow.
Hi, I just want to say that was really cool. I did a project that we like modeled some stuff too, and it looks like that, and it's like super fun to use those programs. So I was just like wondering, like, how did you like, I guess, yeah, like, did you code the proteins? Like, how did those models come about? Did you work on that? Yeah.
So I didn't participate in the actual creation of the model. I did a lot more of the manipulation with the mutational analysis and kind of electrostatics. So I didn't do the actual building. But for anyone who's worked with, so that program is called PyMol. And if anyone is at all interested in structural biology, I definitely recommend playing around with it, just because, especially when you're talking about all of this stuff, it can seem really intangible. And then to say, oh, I have this protein, I can like mess with these residues, and this is how it binds to things. I feel like it's a really cool way to understand biology in just kind of a different lens.
Yeah, totally.
But it is a lot of coding. That's how it's, PyMol works in Python. So
cool.
Yeah, so for any of you coders.
I got time. One quick question. Otherwise, we'll move on. Anyone else? All right. So let's all thank Ella, and well, I guess virtually. Normally we'd all clap. It doesn't work so well now. But I'd like to have Dr. Shellman come to the lectern, as it were, and introduce Chiara Dart.
Hi, I'm Yiqun Shellman, Associate Professor in Dermatology. My lab study cancer and aging of pigment-producing cells. And this program always shows excellent students, as this year we have a great pleasure to have Chiara and Dart in our lab. And she has been very productive this summer, even with the challenge. So the very first half the internship, she actually helped us preparing a manuscript, making figures and editing the manuscript. And I'm happy to announce the paper is accepted this month. So she's the co-author on a paper already. And then the second half, she actually got the permission to come into the lab, and she worked on continue a project she started last summer at my collaborator's lab, Dr. Robinson's lab, and she actually did some really cool experiment with InQSite, and so she will share some of the progress on this project today. Chiara?
Perfect. Thank you so much for that introduction. Let me share my screen. Can everyone see that? Okay. My name is Chiara Dart. This summer I've been working on a project studying the potential therapeutic combination of 5-azacitidine and BH3 mimetics in melanoma. So for some background on melanoma, currently there are 2 standards of care, which are targeted therapies and immunotherapies, and these have really revolutionized the treatment of melanoma. However, there's still issue with resistance, lack of response, or lack of a targetable mutation in certain patients. This is particularly true of patients with rare melanomas, which are this mucosal, acral, and uveal subtypes that you can see over here on the right. So in this project, we looked at a combination of azacitidine plus ABT-199, which is a combination that was recently approved for the treatment of AML in elderly patients. This is one of the studies that they published out of that work over here, and you can see that in this survival plot, this combination, venetoclax and ABT-199 are the same drug. This combination has a substantial improvement over the control of every other treatment that a patient could get at the university. So our goal was to look at azacitidine in combination with a handful of different BH3 mimetics to see if this combination had similar efficacy in melanoma, particularly for these difficult to treat melanomas that don't have many other options. So BH3 mimetics rely on this apoptosis pathway for their action. There are two pathways for apoptosis, this extrinsic pathway over here on the right, which is mediated by proteins on the cellular membrane. We're not going to talk about that a lot. And then there's also the intrinsic pathway, which is controlled by the BCL2 family of proteins. There are both pro and anti-apoptotic members of this protein family. These BH3 only proteins are pro-apoptotic, whereas there's a handful of anti-apoptotic members that they interact with. And ultimately, these interactions lead to the activation of BAX and BAC, which then oligomerize on the mitochondrial outer membrane, release cytochrome C into the cytosol, which then goes on to activate caspases and lead to the degradation of intercellular material. So the way that BH3 mimetics work is that they are able to competitively bind to these anti-apoptotic BCL2 family members that are shown as these kind of purple goalposts. So they're able to bind these proteins and they displace these green balls, which are our pro-apoptotic BH3 only proteins. And those proteins go on then to activate BAX and BAC and release cytochrome C like we talked about on the last slide. In this project, I worked with a variety of different mimetics. These two over here, S63 and S64, inhibit MCL1. This mimetic S64 is a clinical grade version of S63, so we wanted to look both at this drug that was available when we were first starting this project, as well as the clinical grade version that we got our hands on a little bit further down the road, just to see what the drug that you would actually be able to use in humans, what the effect of that was. I also worked with A133, which targets BCL-XL, ABT-199, which targets BCL2, and then ABT-263, which is a pan-BCL2 inhibitor of BCL-XL, BCL2, and BCLW. So the other drug in the combination, 5-azacitidine, is a hypomethylating agent. It is an analog of cytidine. It's able to incorporate into DNA and aid in the degradation of DNA methyltransferase. It has historically, as a single agent, been the standard of care for AML, and more recently has been improved in combination with ABT-199 for treatment of elderly patients with AML, and that has been wildly successful in the clinic in that setting. So one important subset of melanoma cells to target are these melanoma initiating cells, which are a form of cancer stem cell. They are able to divide asymmetrically into more melanoma initiating cells as well as into bulk tumor cells. And it's important to target these cells with your therapeutic because they are able to lead to relapse, as you can see over here on this diagram on the right. Therapeutics that don't effectively target these cells, eventually they will form new bulk tumor cells and your tumor will recur, which obviously is not something you're looking for. So I completed 3 main techniques that I'll be talking about in this talk. The first is a cell viability or ATP assay. Pretty much you add a reagent to a well full of drugs that you're interested in looking at, and it will react with ATP and create a fluorescent signal, which is then read by a reader. And that signal is directly proportional to the number of cells that you have in the well. So it's a way of telling how many cells you effectively killed with your treatment. The second technique is this IncuCyte live cell analysis machine, which is really interesting. It takes photos of your cell at designated time periods that you choose, and it uses both a phase channel, so you can look at just the raw confluence of cells, as well as a fluorescent channel, and you add a dye into your wells to look at a protein of interest. In our case, we looked at cleaved caspase, which is an indicator of apoptosis. And then finally, we completed some sphere assays, which is a way of measuring the effect of our treatment on these melanoma initiating cells that we're interested in, because they are able to form spheres in serum-free media, whereas the bulk tumor cells are not able to do that. So first off, here's some data from an ATP assay. It is normalized back to a DMSO control well, which was set at 100%. So this is just the number of cells alive versus what we would consider to be all of them alive. And these first two over here are single agent treatments. This is azacitidine. This is going to be the same for all the graphs, so I'm just going to talk about it once. And this is our S63, which is again an MCL1 inhibitor. We can see that azacitidine as a single agent has very limited impact on these lines. S63 has a slightly higher impact. There are a couple lines over here which respond with really high sensitivity to the single agent, but overall most cell lines are not responsive at this dose. However, the combination, the 12 out of 14 cell lines respond with a viability of less than 50%, which is pretty good. In the next combination, we looked at A133, which is a BCL-XL inhibitor. And again, the single agent has a limited impact. The combination has more effect with about half of the cell lines responding. However, there's one cell line that actually grows with this treatment. And in comparison to the S63, this is generally a more weak response. The ABT263 pan-BCL2 inhibitor plus azacitidine combination, again, very limited response as a single agent. With the combination, about half of cell lines are responding, but not as impressive as what we saw previously. And then ABT199 plus azacitidine, which is the combination that is approved for the treatment of AML, we saw very little with the single agent. And then with the combination, there's a couple cell lines that respond, but there's also several cell lines that grow with this combination, which was not what we were expecting to see. And based off of this data, we decided to move forward with the S63-845 plus azacitidine combination because it had the highest effect, and we started looking at what might be the mechanism behind the cell reduction that we're seeing. So in order to do that, we used the IncuCyte machine, and we found that looking at proliferation in these two cell lines, these are both uveal melanoma cell lines, in case you're interested in that, and we dosed them at slightly lower than what we were looking at on the last slide because they tend to be more sensitive to single agent MCL1 inhibition. And we found that with the combination, the relative proliferation was substantially decreased for both these cell lines, although slightly more striking for MP46. And then we also looked at a cutaneous melanoma cell line, this SKMEL28, which is dosed higher because it does not have that high sensitivity to the single agent. And we also found a substantially different relative proliferation with these combination treatments. We also looked at, for those same cell lines, the amount of active caspase, which is a measure of the number of apoptotic cells that you have. And we found for the combination treatments in blue and purple, for these uveal melanoma cell lines, substantially increased amount of area with activated caspase. Although we also see with MP41, the single agents having some effect on the level, it is not quite to the extent as the combination treatment. And again, we looked at this cutaneous melanoma cell line and saw the same thing with the combination treatment, increasing the amount of activated caspase in these cells. So the last assay that we were able to run was our sphere formation assay. And we found that this combination of S63 plus AZA, this is again relative to DMSO treated wells that are set at 100%. We found that the combination of S63 plus azacitidine substantially decreases the number of spheres towards around 20%. And there's over here on the right some representative images of the spheres. And obviously in this DMSO well, we see larger and also more spheres. And then when we look at the combination well, they are pretty annihilated. So in conclusion, the single agent treatment with BH3 mimetics or azacitidine doesn't have much of an impact. The combination of S63 plus azacitidine is effective in the majority of the cell lines that we looked at. And this AZA plus MCL1 inhibition combination is able to decrease proliferation, increase the number of apoptotic cells, as well as decrease sphere formation. And just one main take home for this is that the combination we're looking at of MCL1 inhibition plus azacitidine is potentially a therapeutic option that might be effective for patients with melanoma, specifically these rare melanoma subtypes that I talked about in the beginning. I would like to thank members of the Shellman lab, Dr. Shellman for mentoring me through this process, as well as Nabanita, who has taught me a bunch of techniques and always been available for questions. Also, many members of the Robinson lab. Dr. Robinson also played a large role in mentoring me. Carol has always been available to help me with troubleshooting data or to bounce ideas off of. And then Dr. Casey Couts, Rob, and Morgan for helping me with cell lines and also answering any questions. And of course, thank you to the benefactors of the GSIP internship for making my work possible.
All right, thank you very much. Great job, Chiara. We have time for a few questions again. I'd like to solicit them mostly from the interns first, if we have them. Just go ahead and unmute and ask your question. I'll ask a question then in the meantime. Do you think this combination of drugs would influence tumor bulk or metastasis, or both?
So we didn't do any sort of like migration work. I think based off of what we have done, it's most likely that it would impact tumor bulk. I can't really speak to the impact it would have on metastasis based off of the data we've gathered right now.
Thanks. We have some other questions. Mark?
Yeah, that was a great talk. Congratulations on the beautiful study. I'm just kind of curious, would these drugs be oral drugs or something that topically could be put onto, say, a skin melanoma?
So I don't think either of them are available topically. I believe azacitidine has recently become available orally in the S63. I'm not entirely sure how they're administering it. It's not currently clinically approved, but it is in a couple clinical trials at the moment.
Yeah, okay, thank you.
Do you know, or does your lab have any data on how the drugs affect parts of the body that aren't melanoma? You know.
So I have not done work on that. Through other studies, the lab has done work in animals that looked at toxicity, not for this combination, but for a couple of BH3 mimetics in combination with each other. And those, you can achieve tumor reduction at levels that aren't toxic to the mouse model, but I can't speak to specific areas of the body.
Cool. Thanks.
All right, let's all thank Chiara. Great job. And